presentation. Back in 2020, we hosted a virtual symposium. And at that time, we had a discussion. We talked about, I remember Dr. Angie Serrano presenting about generation of patient-specific iPSCs and cortical organoids and biobank of iPSCs. And a year into the conversation, we had children and siblings who participated in this research and provided their samples at the June 4th conference. So it's very fascinating. It's a proud moment for us today as we continue to dig deeper and better understand the world of iPSCs. And to support this endeavor, we have Dr. Gustavo today. Dr. Gustavo Mostoslavsky is the founder and co-director of the Center for Regenerative Medicine at Boston University. Dr. Mostoslavsky has a broad research background in medicine and molecular and cellular biology with specific training in nuclear programming, reprogramming, pluripotency, stem cell biology, and gastrointestinal specification. Dr. Mostoslavsky devoted significant efforts to the development of a methodology for relevant iPSC and their use for human disease modeling. And it's amazing to see that these iPSCs developed at CREM have been freely distributed for use in more than 700 laboratories across the world. Wow, that's amazing. Thank you for joining, Dr. Mostoslavsky. You may start sharing your screen.
Well, hello, everyone. Let me put my slides on. There you go. So I'd like to thank the foundation first, and of course, Dr. Angie Serrano. She put me in touch with the foundation. I've been hearing more and more about KAT6. I'm not going to be talking about KAT6 yet because I'm going to leave that to Angie actually to do all the work on those specific samples. But I'm just thankful that I have the opportunity to tell you about the work that my laboratory does and the Center for Regenerative Medicine. And I like this joke because I believe that actually it's not a joke at all. I believe that a lot of families with individuals that suffer from rare diseases, this is the feeling they have, that really every time they hear about a potential cure, it feels like it's a light years away. And so I hope I can convince you today that we are developing the very powerful novel platforms that we believe will be very useful to study the diseases, these rare diseases, and hopefully the cure will be actually much sooner than we think. And this is the building where we are, the Center for Regenerative Medicine. The second floor in this building is where the center is. And a lot of the facilities that we have are centered around culturing cells in vitro. This is a typical tissue culture room that we call. We have actually 5 of these tissue culture rooms now in the center. And a lot of the work that we do is centralized around this very particular type of stem cells called iPSCs, or induced pluripotent stem cells. I do need to apologize to the scientists in the audience because I decided to put a few very basic slides to give the families the opportunity to understand better what are these concepts when we talk about stem cells and pluripotent stem cells. And this particular case of pluripotent stem cells starts actually almost 4 decades ago with the discovery of a type of stem cell called embryonic stem cell. And a couple of researchers group found a way to capture a fertilized egg. They cultured them for a few days in vitro, and they formed this structure called blastocyst. And it took really a lot of work to find conditions where they could take the cells that will become an entire organism, an entire embryo, and put them in vitro. And they start growing as colonies, and that's why they're called embryonic stem cell colonies. And the fascinating aspect of this cell, and that's why even almost 40 years ago, it caught really a lot of people's attention, was that at any time point, you can keep these cells growing in vitro forever, literally for years. But if you change the conditions in vitro and you put certain goodies, certain growth factors in vitro, they actually have the power to become every single cell lineage or tissue or organ in our bodies. Now, there were a lot of technical issues related to embryonic stem cells. So for years, scientists dreamt about the possibility of making these embryonic stem cells, but not actually by destroying embryos or by using eggs, which was complicated in the case of human cells. And in 2006, there was a major discovery by a Japanese scientist named Shinya Yamanaka, who found a way, a methodology in which he could take, in that case, was skin fibroblasts. And the first paper was from mice, but a year later they showed that the same thing works for human cells. And by forcing expression of 4 transcription factors, 4 genes basically, which was something, by the way, at the time no one believed that would be possible. These cells change their identity. They really almost go back in time in a process called nuclear reprogramming. You are really changing the way that the nucleus of those cells express genes, and they start making these colonies of pluripotent stem cells that look basically identical to embryonic stem cells in many regards, and they call them induced pluripotent stem cells, or iPSCs. Now, you don't need to take my word for it. How do we actually know that these cells are pluripotent? And really the ultimate test, it's a functional test, and we cannot do that with human cells, so we use mouse cells to prove that. And the way that we do it, we take these iPS cells and we actually inject them back into a blastocyst, the same blastocyst I mentioned before, and we do a trick where our iPS cells come from a mouse that is either black or dark brown. It's called agouti, and we put it in a blastocyst of a donor mom that is white, it has a white coat. So then we just let mice to be born, and let's see what happened. And biology works. So this is a chimeric mouse. And every time you see here a dark patch, a dark part of the coat, those cells must come from the iPS cells we put in the blastocyst. But even more powerful, if the cells are truly pluripotent, they should contribute not only to skin, but to all the tissues and organs of these animals. So we can take now one of these animals, and if there is contribution to the germ cells, we can breed this animal with a white mouse again. And if these cells are truly pluripotent, we should expect now mice that are entirely either black or agouti or white. And this is indeed what happened. So the fascinating aspect, I need to tell this again, we started with a little bit of a piece of a tail, skin fibroblast, or we can now routinely do it from a little bit of blood. And from those cells, you can now have an entire animal completely coming out of those cells. So this is a very powerful technology. Dr. Yamanaka actually got the Nobel Prize for his discoveries only 6 years after the original discovery. I was lucky to meet a few times Dr. Yamanaka in different meetings. This one is in Barcelona. And why all the fuss, why all the noise about iPSCs? Well, this is the first time actually in biomedical research that we have the ability to generate pluripotent stem cells from any individual, because pluripotent stem cells are not in our bodies. We don't have any more pluripotent stem cells. So this platform allows us for the first time now actually access iPSCs, or pluripotent stem cells, from anyone. And you can imagine that, and we do this routinely, as I said, for a few mLs of peripheral blood. And we can now make the cells differentiate. This is called directed differentiation, where we actually direct the cells to become a cell type, a cell-specific lineage that we are interested in. And you can imagine there are many usages for those cell lineages and tissues, and one major one that we use is disease modeling. And one day we hope that we can also use the cells for cell therapy and replacement. And you can imagine that inside this box, of course, we can also put KAT6 because it's, like I said, very easy now for us to just make iPSCs from any individual, which is what Dr. Serrano is actually doing, and she collected the cells already to start making iPSCs from individuals with the KAT6 mutation, many from your families. So my lab does a lot of things. That is true. We do have human iPS as a centralized theme, but we go into many different directions, which I think also prove the very amazing power of these cells, that we can make hepatic cells. We're making a lot of different type of blood cells, like lymphocytes and macrophages and dendritic cells. We have a very exciting project studying a familiar genetic disease called Creutzfeldt-Jakob disease, caused by prions. So we're making neurons and brain organoids from these iPSCs to study the disease in vitro. But the topic of today, which is related to the webinar, is our ability to make something we call intestinal organoids. And we've been using this intestinal organoid for a lot of things. So I'm going to tell you today a story that we published a couple of years ago, trying again to convince you that why we believe that this is a very powerful technology. And we'll give you also a couple of examples of how we are using this platform. So the first part is just to tell you about our study on a new way of making these intestinal organoids from human iPSCs. And the story started, it was completely done by a very talented MD PhD student I have in my lab, Aditya Mittal, who's back now in medical school at BU. And we were using another protocol for intestinal differentiation that we were not very happy. And he noticed in another protocol that was used at the Center for Regenerative Medicine to try to make lung cells, that in that protocol, actually, after a couple of weeks, there was an emergence of a significant number of cells that express a marker called CDX2. And CDX2 is a very early marker that actually identify in endodermal cells early hindgut. And so he thought, well, maybe this protocol that people are trying to use to get lung may be also useful to get gut. So he put a lot of effort and focus on trying to develop that protocol. And indeed, he tried a condition first that was known just to mature lung organoids, but choosing cells that were negative for markers that will be lung. And when he did that, he obtained robust proliferation of these, what we call 3D organoids. So we didn't know at that time they were intestinal or not. But indeed, he did a few stainings for markers that classic of the intestinal epithelium, including CDX2, as I mentioned, which is a transcription factor, but also villi, which is very strongly expressed in the villi part of the gut. And indeed, all these 3D structure express very strongly these markers. But even more importantly, we did RNA sequencing on these organoids and comparing the protocol that's supposed to give rise to lung. And you can see here, all these in red are genes that are upregulated, in blue, genes that are downregulated. And you can see that most of the genes that are red are genes identified very specifically in lung cells, like surfactant protein cell genes. And indeed, the computer, if you ask, okay, what is the number one tissue identified with this type of signature, it is lung. In contrast, when we did our protocol, almost all of these genes that are upregulated in red are typical genes expressed in the intestinal epithelium. And this is a lung gene that is not expressed. And indeed, again, if you ask the computer to tell you what is the tissue that is mostly identified with this type of signature, the number 1 and 2 was colon and small intestine. So we were very happy with that, but he was not happy. He said that, we need to improve it and we need to see if we can find even better conditions. So I'm showing here an example of 9 different conditions he tested to improve on this differentiation. And I want you to focus only on 2 of them. One is this media. I will call it IMCK from now on. And not only the cells grew very, very nicely, and you can see this here by quantifying the number of organoids that we get in each differentiation, but also they robustly express different markers of intestinal that I'm not showing here. I'm going to show in the next few slides. And we did all of this comparing to the classic media that we've been using before, CK-DCI, and we started trying to identify if there were differences between these 2 types of conditions that we have to grow our organoids. And I would like to you to focus on a couple of things. So here what we're doing, we are analyzing expression of genes by just a technique called real-time PCR. And I want you to focus on first this gene and this gene. So this is mucin2 and this is SATB2. And these are genes that are very highly expressed in the colon, but not in small intestine. And what you can see is in the CK-DCI condition, and these genes are very, very highly expressed, in contrast to the IMCK condition, which are almost not expressed at all. Strongly suggesting that this CK-DCI condition, it tends to make the differentiation of what we will call distal colonic-like organoids. And in contrast, the IMCK condition tend to differentiate intestinal organoids that represent more the small intestine. And an example of this is the expression of this gene PDX1, which is not only pancreatic, but also very highly expressed by duodenum, which is part of the small intestine. And it's the same thing is for CDH17, which is expressed in both in colon and small intestine, but much higher in small intestine. So we actually were very fortunate, I guess, that Aditya was able to find conditions that allow us now to specifically differentiate intestinal organoids that have regional identity, either small intestine or colonic. And this has a lot of clinical relevance because there are several diseases that affect specifically the colon and not the small intestine, and vice versa. We did a few more characterizations. So here where you can see very robust secretion of mucin into the lumen of this intestinal organoid. So also supporting the fact that these organoids are functional. And we also did staining for other important cell types that's supposed to be present in the gut. One of them are lysozyme expressing Paneth cells, and the other one are enteroendocrine cells, in this case expressing chromogranin A, which is an enteroendocrine enzyme. Now, the final part of the characterization of these intestinal organoids, which we believe was another advantage of compared to previous protocols, is that our organoids grow completely mesenchyme-free. And what that means is that in previous protocol, there was a very strong need for the presence of a mesenchymal cell support. And you can see that by here, when we do stainings against an epithelial cell, EPCAM, it's a marker of epithelial cells. You can see here that more than half of the cells that do not express this epithelium strongly showing that the presence of these mesenchymal cells. In our protocol, they are completely gone. Actually, our organoids grow completely devoid of mesenchyma. We can even see that, of course, in the microscope. And this is important because for several applications, if you want to study epithelial only cell intrinsic effects, not having mesenchyme, it is important. In that publication, we also reported for the first time the creation of a new reporter line. And this is important because for many laboratories, having access to iPS cells that have the reporter, specific reporter, can be key in how they study a specific lineage. In this case, we generated using CRISPR-Cas9 gene editing, the introduction of a GFP reporter into the CDX2 locus. And that means that if the cells express CDX2, they will also supposed to be green. And this allows us now to actually sort, to identify those cells that very early will express CDX2, and those will be the cells that we believe will become the intestinal organoids. And indeed, when we did first differentiation into endoderm, this is the first step in the differentiation protocol, very high efficiency of differentiation. And then we can see and follow the differentiation for the expression of GFP, the appearance of GFP. And we can see by day 8 already that almost 35% of the cells are expressing GFP, and even more by day 13. So we can sort those cells out based on green, and we let them grow. And of course, they become beautiful organoids that are all green. So these have now become a very robust and efficient method for us to make these intestinal organoids in vitro. So I'm going to give you, so what can we do with these intestinal organoids? I'm going to give you a couple of examples. One has to do of course, with SARS-CoV-2, when the pandemic hit, many labs needed to close, except if you were working, actually doing some work that is called essential work related to COVID-19. So we decided to take advantage of our platform to see if we can study the infection of SARS-CoV-2 in the gut. And the reason, the rationale behind it is that actually more than 50% of patients that develop COVID-19 at the beginning of the pandemic had clear gastrointestinal symptoms. And as a matter of fact, there are a few anecdotal reports showing that in feces, in stool, you can detect the virus even much longer after the respiratory samples have become negative. Even more important for us from a mechanistic point of view, there were a lot of controversy about whether having an infection in the gut was beneficial or not for the disease. And the claim was that maybe the virus was stimulating some type of inflammatory response that could be beneficial, while other groups have claimed that it was the opposite, that the inflammatory response was making the symptoms even worse, and people with gastrointestinal symptoms were staying even longer in the hospital. So we wanted to see if we can use our platform to maybe shed some light on that mechanism. So we started a collaboration with a great friend and colleague of mine, Dr. Elke Mühlberger. And Elke works in this building called the Needle, or the National Emerging Infectious Disease Laboratories, which is across the street from our building. It's also part of BU. And it's a very special building because it's one of 3 buildings in our whole America that can actually do work which is called BSL-4. So they have containment to work with all kinds of nasty and dangerous viruses. And because of that, they were actually the first building to receive a permission and actually to receive the live SARS-CoV-2 virus in Hall, Massachusetts. So we were very rapidly, we could have access to this actual live SARS-CoV-2. So let me share with you the data that we published also last year. So we made the proximal and distal intestinal organoids, and we exposed them for a few hours to SARS-CoV-2, and then we perform stainings and see, okay, are the viruses infecting the gut, the intestinal organoids, and is that happen or not actively. And what you can see here in red, this is a staining against the N protein of SARS-CoV-2. So every time you see red, it's actually presence of a viral particle, and I hope you will believe me that you can see that the infection was very, very robust. This is 4 days post-infection, both in proximal and colonic, and the colon seems to be even more sensitive to infection. But as a matter of fact, when we checked by real-time PCR, which was the same PCR we developed to help the hospital diagnose COVID in patients, we can see very robust that even after day 1, we can detect the presence of both N1 and N2 protein from the, in this case, genes from the viruses in these samples, both in proximal and colonic. And even more important, the cell seems to provide a strong support for replication. This was something that was completely unknown in the field. Do SARS-CoV-2 viruses can actually replicate or not in the gut? And based on this experiment, we are measuring titers of viruses extracted from these organoids by day 1 and day 3, and we can see a significant increase in these titers, strongly suggesting that the viruses are replicating. But even more important, I told my student, well, you know, you show me a red dot in this organoid, that's not a virus. I want to see the virus. So the only way you can see a virus, of course, is by doing electron microscopy. So we did electron microscopy on our organoids, proximal and distal, and we saw the classic picture of, you know, this crown surrounded the surface of the spike protein that became very popular. Everyone knows the word spike now, thanks to this SARS-CoV-2. So a lot of robust presence of these viral particles in the organoids, and not only in both proximal and colonic and distal, but also the formation of this very specific structure, monomembrane and dual bilayer horseshoe structure that are actually associated with previous coronavirus infections that are known to represent replication complexes for coronaviruses in cells. So if we do now, again, RNA sequencing, the first thing that is very obvious here in circles, you can see the colonic organoids, in triangle, the proximal ones, and we compare mock infection to infected cells, either at day 1 or day 4. Day 1 is green, day 4 is orange. And what you can see, these are 4 different genes of the virus. Very clearly, not surprisingly, we see a dramatic upregulation of the expression of these viral genes in day 1, and even higher day 4, again, strongly supporting the fact that these viruses are replicating. But if we ask the computer to tell us a more sophisticated analysis of the RNA sequencing, how many genes are differentially expressed between uninfected cells to infected cells at 1 day and 4 days, and you can see here that at 1 day we see the appearance of these genes, and these are 600 genes in the proximal, only a few genes, 60 genes in colonic, but much more robust differences at 4 days post-infection. So what are these genes that are differentially expressed? So again, we asked the computer to show us in a heat map how what are those genes. Surprisingly for us, we saw that mostly in the colonic organoids and not in the proximal, there were robust expression of a group of genes that are called iPSCs. These are interferon signaling genes that are related very strongly to interferon responses, which is actually a response that a tissue will show if it's infected with virus, but it's not that common to see in epithelium. So here we show that the gut was responding to the infection. Here a few examples of these genes: IFID1, IFID3, CXCL10, which is a cytokine receptor, and a few others. And we have other ways of analyzing this data. So we did a GSEA, gene set enrichment analysis, which aggregates a group of genes in what is called hallmark responses. And again, the same type of things. We see here in the bottom, you can see all the interferon responses. And also we see some actually strong appearance of inflammatory responses upon infection with the virus. So every time, again, you see a triangle, the triangle means significant and the red is upregulated, blue is downregulated. Even probably more significant for us was that when we try to compare this type of signature with other type of RNA-seq data from other lineages that people have done before. So we compare it to a couple of databases from adult intestinal stem cell derived organoids. So these are organoids actually derived from the gut itself, or to a cell line, an intestinal cell line called KAT6, or our own database from the CREM analyzing infection of lung organoids. There is a lot of commonalities on this type of inflammatory response that we observe, strongly suggesting that many of the phenotype of the clinical phenotype these patients experience could be targeted using drugs that target these particular pathways. So in conclusion, for this part of the studies, we show that our intestinal organoids derived from human iPSCs support productive SARS-CoV-2 infection replication. Both proximal and colonic organoids show the typical osteostructural changes associated with viral assembly and replication. And we found that the infection with SARS-CoV-2 elicited robust, but it was a delayed transcriptional change. And we believe that that may explain why up to 50% of the patients actually experience significant gastrointestinal symptoms. And importantly, this transcriptional signature appeared to be common to other epithelial lineages, including primary gut and lung cells. So we hope that this can help illuminate novel therapies for this type of infections. I didn't have time to show, but we also observe upregulation of cell death-related genes, strongly suggesting that the infection with the virus is also causing a significant injury in the organoids. So we hope that what else can we do? So this is just another example of another project currently ongoing in our lab. So we also had a collaboration with KAT6 to study Ebola virus. This is another type of very nasty virus, and patients with Ebola infection, up to 60% will die in a matter of days. And one of the main causes of death is a massive loss of fluids from the gut. And no one knows anything about actually Ebola and viral infection and intestinal infection. So we're using this platform to actually infect. We already did infection. Here you can see, again, our intestinal organoids infected with two types of viruses. One is Ebola virus, and another is a similar virus called Marburg virus. It's also very nasty pathogenic virus, but it has a different clinic. So for us, it's like a control. And again, in red is the detection of Ebola virus in our intestinal organoids, and we can see a very strong infection. And we have started analyzing by RNA sequencing what is happening to these organoids when you infect them with Ebola virus. So what else can we do with these cells? Well, we actually have been using intestinal organoids to study genetic diseases. So we have high hopes that now that Angie has collected the samples and we're going to start reprogramming them, we'll be able also to use this platform to study KAT6A in the context of intestinal problems. But this is an example of another genetic disease that has mutations in a gene called iPSC, which is strongly associated with the development of a disease called familial anastomosis polyposis and the development of colon cancer. So we published an article showing what happens if you mutate iPSC in iPSCs and then make intestinal organoids. And another type of thing that we believe will be closer to the type of tissue engineer and that we envision one day will be possible to do. We have a collaboration with a group in MGH in Massachusetts General Hospital led by Dr. Harold Ott, who is a surgeon, and he developed a technique where he can take a piece of an organ. In this case, this is a piece of a rat gut, and we can clean all the cells out of that tissue. It's called decellularization, so that you end up with the perfect scaffold of the proteins that actually make the tissue itself, but no cells. So with that, now we can recellularize that piece of tissue with our own iPSC-derived intestinal cells. And what we were able to show in this paper that the cells actually make beautiful epithelium, almost like a normal gut, and it was actually functional. So these are a few examples of where we think this field is moving. I want to end up, of course, with the most important slide to acknowledge the people that did the work. Most of what I showed was done, as I said, by Aditya, who just graduated from his PhD, and now he's back in finishing his MD part. These are some of the funding that support the the lab, and I'll be happy to take any questions.
Thank you so much, Dr. Mostoslavsky, for explaining a complex science. So if you have any questions, you can also put it over the chat. All right, so I have a question, and this is about the iPSCs in general. How viable are these iPSCs, like in terms of families? They provided blood samples at the conference and the precious iPSCs were developed. Do they have to do this over and over again?
No, the beauty of this, that's why there are stem cells. So the moment you have, you can extract a little bit of blood and freeze it actually. And then you can thaw whenever you have the funds and the time and the resources, you take the blood that is frozen, you thaw the cells, the blood, and you do what is called nuclear reprogramming, which is a process that takes around a couple of months to then have these colonies of iPS cells. So the cells of the blood basically, as I mentioned before, go through this process of nuclear reprogramming to become this induced pluripotent stem cells. And once you make the pluripotent stem cells, the iPSCs, you can basically expand them indefinitely and make a bank of vials of these cells that you can freeze. And these cells actually can grow forever, literally forever. I can give you a couple of examples from both sides. You can do that even with cells that have been frozen blood for 30 years, which I did in one of my projects for the intestinal colon cancer. We actually did an analysis on a large family with Lynch syndrome, which is another cause of colorectal cancer. And most of these patients had been dead. And we were able to take peripheral blood that was frozen from this individual that had been dead for many years and make these pluripotent stem cells that are actually pretty amazing, and that you can now freeze and have in the freezer or keep them growing and making organoids indefinitely.
Okay, and we know that from Angie's work that there's a methodology for creating cortical organoids. I want to understand, and you mentioned from the other work that you have been doing about the intestinal organoids. And again, I don't have a genetics background, but I just wanted to know if the gene variation can come in way of the methodology, and it will change the methodology or play with its robustness.
It's a great question. In general, we believe that if the gene doesn't affect development, meaning if the development of the individual is relatively normal, we expect the cells to behave normally. And the hope is that at the moment, by the time you obtain what we like to believe is closely related, more mature intestinal organoid or brain cortical organoid, you can start seeing the effects of having the mutation. Now, in the particular case of the Angie, and I know I'm going to let Angie explain that better, I believe that she actually was able to find some differences even very early when she was studying the brain organ. So it depends on what the phenotype is of the gene. It could be an effect on the organoids much earlier. I think it depends on what's happening, of course, in the patients. But I don't know if Angie, I see Angie desperate there to talk.
Actually, I'm desperate to ask you a question, Gustavo.
Okay.
Let me follow up with what you say. Thank you, KAT6 Foundation. I love the new logo, by the way, and Gustavo for sharing your expertise with our community. Yes, it's in our case for the other disorder I'm studying, Kabuki syndrome, we do find a difference that we believe might be affecting a little bit our differentiation protocol for neuroepithelium, but that's something, that's preliminary data, and that's something that we're actually looking into. And I think that it's great that the community is paying attention to this lately. And Gustavo, I have like two quick questions. One of them is just out of curiosity. I could not stop observing that you have the vimentin gene downregulated in your RNA-seq data for intestinal organoids. And I was just curious why that was the only gene highlighted. But
because that was very specific, because vimentin is very highly expressed in the mesenchyme. So for us, it was very important to show that we didn't have any mesenchyme. So that was a very targeted gene. That's why it was there.
Oh,
so it was
thank
another proof. So it has to do with actually I know why you are asking that, but in this particular case for the intestinal organoid protocol, it was very important as to convince because it was actually very controversial that we could actually grow the organoids completely mesenchyme free, because it's very different from how people have been doing this differentiation before. So it was very important for us to convince people that actually the cells are entirely epithelial organoids and no mesenchyme.
Wonderful. And the second question, probably more for our community, you show wonderful examples of how you were measuring the changes in gene expression or the changes in these organoids because of viral infections. So I was wondering if there is any other kind of phenotypic analysis that you could do that might help us to correlate sometimes disorders, GI disorders in patients with brain organoids. What kind of things can you measure in intestinal organoids?
Okay, so there are several things. Of course, when you do RNA-seq, you're basically measuring everything, right? You have a very broad knowledge of what is the genetic signature of these intestinal organoids. And of course, if you have the ability to compare mutant versus not mutant organoids, and that will give you a pretty, I would say, pretty robust broad picture of what can be wrong. But of course, that is at the level of gene expression. But you can imagine like how very easily you could translate that in, for instance, identifying what could potentially be biomarkers. So I am involved in this Kreutzfeldt-Jakob disease modeling. We're very strongly trying to help also a foundation, CJD Foundation in Israel, to find the biomarkers of this disease, which is something that it doesn't exist. There are no biomarkers right now to identify why a patient will develop the disease at all. So I could imagine that for KAT6 could be something similar. You could start thinking about, okay, if we have now this intestinal organs with the mutation, without the mutation, you make as fully mature as you can. You can test not only gene expression, you can start also doing some very basic functional analysis. And this functional analysis is based on what we know about the gut. So basically there are, the very obvious one is absorption of liquids or fluids. So you can actually measure, you can use a technique that relates to what's called a transwell. And you can actually measure entrance or exit of liquid or fluids into these organs. That's one way of doing it. Or you can actually measure the function of what we call ion channel that is the epithelial of the gut express. So there are several different ion channels that the gut epithelium express that control the entrance and exit of ions, salts, and different molecules and liquid. So you can directly test the function of that. So that's another way of testing basically the function of the intestinal organ. So you could argue that that would be another thing you could test to see if it can help you find correlation between that and some of the clinical the gastrointestinal clinical phenotype of the patient.
That's wonderful. Thank you, Gustavo.
Thank you so much, Dr. Mostoslavsky. I'll just ask one more question. It's from the chat. For the nuclear reprogramming, does the blood have to come from a direct biofamily member, or can it be from any of the kids' KAT6 family members?
Well, that's a great question in general, and I know that Angie already organized some of that collections. In general, when we design these studies, we try to be as broad as possible, and it's very important for us to have access to family members. That's the first thing, and I will explain why. And also family members that are carriers versus not carriers of the mutation. And the reason behind it is because human beings are very, very variable. And when we make iPSCs, even iPSCs from different human beings, they can have a lot of differences just because of the variability of between human beings. So having access to samples that come from, to iPSCs that come from people that are genetically related, like family members, it's a big plus when you build a library. And that's why when Angie collected, when I collect samples to make a bank, to make a library, it's very important that we have access to as many, for as many individuals as possible, and second, to have both carriers and non-carriers. So I guess that the answer to that question is no, it doesn't need to be, because we also need access to non-carriers, but having access to family members makes the bank more valuable.
Okay. Thank you so much for your time. Thank you. We really appreciate it. So we'll move on to our next presentation.